Hello, and thank you for joining me today. I am Jacob Hogins, and I'm going to be going over our research on to pool or not to pool, a comparison of bacterial growth characteristics in pooled and individual urines. We have no affiliations or funding sources to disclose, so we're going to just skip over that. Researching UTIs in vitro pose many problems that compound and confound the results of the studies. The means by which these studies are conducted differ from lab to lab, and whether bacterial cells are added at a certain density or if overnight cultures are simply diluted. And further, the lengths of times in which these experiments are ran varies between labs. These factors, in combination with facts that no two labs can have the same urine samples in pools, and the collection requirements differ between labs, makes reproducing a study nearly impossible. The requirements for our study were postmenopausal non-diabetic women above 50. These urines were then subdivided into patients who have a history of and current UTI, patients who have a history of but no current infection, denoted as history, and patients who have reported never having a UTI. These urines were filtered through a 2.2 micron filter to remove contaminants, and equal amounts of the individual urines were added to a conical vial. Both of the individual and pooled urines were frozen at -80 until further use. The following curves represent the growth of LRPF007, a group B2 uropathogen isolated in the Reitzer lab. The green curves, or curves with diamonds as points, show the pooled urine growth. For the current urine, the pooled urine is neither the average, best, or worst urine. It does slightly better than the majority, but is worse than the best urine, which was urine 1. For the history urines, the initial growth is equivalent to slightly greater than the average, but the dynamics of the curve are different from the final growth. This thus led us to believe that the history urine pool is better than the average growth for the rest of the urines. Finally, for the never urines, the growth phase overall, including the final OD, is much greater than that of the next best urines. These trends maintained for both the other UPEC strain assessed, which was UTI-89, and the non-pathogenic E. coli W3110. We can conclude that regardless of the group, the pooled urine is not equivalent to either the average of the urines or the average of the urine that supports the next highest growth. This suggests that the pooled urine is in fact a media representing no patient, or the average, and instead represents a patient that doesn't exist. A ghost patient. The variability within these runs was unexpectedly quite high, so we took this to believe that this is the variability seen between labs, and we sought to identify the sources or sources of this variability as best we could. Next, we analyzed the ratio of growth between each of the groups of urine. The following graphs show our results, and surprisingly, the growths could be divided into two subgroups that correlated with our UPEC and non-pathogen. The uropathogenic E. coli strains and low density of W3110 grow at a ratio of 3 to 2 to 1 with respect to the current never in history urines, while the non-pathogen at high inoculum grow in a ratio of 7 to 5 to 1 with respect to never current in history urines. This phenomenon can be explained by two different factors. The nutrient content of never urine is likely greater than that of the other two groups, as exemplified by the greater growth in all types of bacteria, while the density dependence is likely due to the innate immune or other inhibitory factors present in never in history urine. Given that the UPEC strains grow better in most types of urine and inoculation density plays less of a role with UPEC, the limiting factor likely is the amount of nutrients present in the urines. If this is the case, then UPEC strains will grow better than the non-pathogens at lower percentages of a nutrient broth known as LB. To assess this, we grew bacteria in decreasing percentages of LB diluted in phosphate buffered saline. And surprisingly, the results aligned with our prediction. The model UPEC strains grew better than the non-pathogen, as exemplified by the decrease in change in optical density of the bacteria after growth in descending amounts of LB. The uropathogens were able to grow well until grown in 5% LB, where the model pathogen UTI-89 began to grow about as well as our non-pathogenic control W3110. This exemplifies that the UPEC strains are in fact able to grow better at lower amounts of nutrients. When pooled, bacterial growth in urine does not exhibit the average growth in the individual urines in that pool. This suggests that pooling urines can shield researchers to the inhibitory elements in one or more urines by compensating with the beneficial elements from another. Nutrient content varies greatly between individuals and within groups, and between groups. Thus, we suspect that elements that affect growth are mainly nutrient-limiting factors as opposed to antimicrobial factors. Expanding on this, the UPEC strains are better at utilizing small amounts of nutrients, suggesting a reasoning for why the UPEC grow better than the non-pathogens. This was exemplified by the growth in decreasing percentages of LB. Though the evidence is still preliminary, we have reason to believe that the reason why the non-pathogens grow better when inoculated at higher densities is due to the fact that they are able to resist antimicrobial peptides at higher densities but are incapable of doing so at lower densities. As medicine moves towards a more personalized approach to treating patients, it would follow that researchers need to move towards assessing the individual factors that affect an infection, especially one that causes 8 million doctor's visits per year. However, pooling is the standard in research, and our data provides evidence that pooling urines diminishes the individuality of the study, yet does not increase the reproducibility between labs. Because of these two factors, it only makes sense that researchers should no longer pool urines, and instead, the development of a synthetic urine should become a main focus. Such a media would be able to be produced in any lab, and can be altered to include factors discovered by assessing individual urines, and yet can drive reproducibility between labs.